every living thing on Earth has a genome made out of the same 4 DNA bases: A, T, C, and G. It's the number and order of those different bases in each organism that gives us all of the variety of life that we see. And even within a population of organisms from a single species, no two organisms are exactly the same. Individuals have different traits as a result of small differences in their DNA sequence. Like here, these two flowers have 2 different colors. And these differences come from small DNA differences in their genomes. These genetic differences make up a population's genetic variation. In this video, we're going to talk about what genetic variation is, how it arises through mutation and sexual reproduction, and why it's important. So let's start by defining genetic variation. Genetic variation is a term that covers differences in gene sequences between organisms. The 2 main sources of genetic variation are sexual reproduction and genetic mutations. Let's talk about mutations first. Mutations are really just typos in the DNA sequence. They can be swapped nucleotides, like a C here instead of an A, or a small insertion or deletion of base pairs. This can happen when cells replicate their genome to make new cells. They make copies of their DNA by carefully pairing each base on one side of a strand of DNA with its complementary base. And this is usually super accurate. But about once in every 10 billion base pairs, a mistake can happen, like this C that was accidentally put in instead of an A. And those mistakes can change the code or expression of genes, which can change a phenotype. When these mutations occur within the coding region of a gene, they can create a new version or allele of that gene. And sometimes the environment can cause mutations too. Things like UV radiation, X-rays, and certain chemicals can damage DNA. When a cell repairs that DNA damage, it can sometimes make a mistake and add one of these small mutations. Mutations that happen in one cell can be passed down to any new cells that result from cell division. But to be passed on to a new generation, like from parent to child, a mutation has to happen in a germline cell, something like a sperm or egg cell. These mutations, if they result in a phenotype and are passed on to a new generation, are what can add heritable genetic variation to a population. The other main source of genetic variation is sexual reproduction. By combining DNA from 2 individuals, alleles get shuffled together, creating new genetic combinations. This rearrangement comes from 2 main processes: meiosis, which happens in each parent individually, and fertilization, which combines the DNA from 2 parents. Let's go over meiosis first. Meiosis is important because it creates genetically unique gametes, like sperm or egg cells, from the genome of an organism. Many organisms, including humans and other mammals, are diploid, meaning that they have 2 copies of every chromosome, 1 from each parent. These are called homologous chromosomes, like this blue chromosome and this yellow chromosome. And while those chromosomes can have the same set of DNA and genes, they can have different alleles of those genes. But for reproduction, each individual gamete must have only 1 copy of each chromosome, making it haploid, so that when 2 gametes come together, the resulting organism is diploid as well. And the process of going from diploid to haploid results from meiosis. Meiosis involves 2 rounds of cell division. Now remember that this blue chromosome here and this yellow chromosome are homologous chromosomes, and each one of those is going to be duplicated in the first part of meiosis. Each individual chromosome is replicated so that the cell now contains 4 copies of each chromosome. Now there are 2 exact replicas of each parental chromosome. The 2 replicas, referred to as sister chromatids, are connected at their centers. And this happens for every chromosome pair in the cell. The matching chromosomes find one another in the cell and line up so that each copy of a chromosome is next to its other copies. So here you can see that the pairs of homologous chromosomes have come together really closely, and they're actually overlapping at some positions. The adjacent chromosomes can swap small pieces of their DNA at these positions in a process called crossing over. This means that a version of a gene that originally came from one parent could now reside on a version of the chromosome primarily from the other parent. So this blue piece of a chromosome can cross over with this yellow piece of a chromosome and swap their DNA so that the resulting chromosomes look like this. This will eventually increase the diversity of genetic variation found in the gametes. Next, the chromosomes align in the center of the nucleus, matching chromosomes side by side. And one set of sister chromatids from each pair of homologous chromosomes is pulled to either side of the cell. Then, when the cell is cleaved in two by cytokinesis, each resulting daughter cell contains a haploid genome but with duplicated copies of each chromosome. In meiosis II, these daughter cells are split one more time without DNA replication. So the chromosomes line up in the center of the cell, and one sister chromatid from each pair is pulled to each side so that when the cell splits in two again, the resulting cells each contain a single haploid genome with one copy of every chromosome. And as you can see from our example here, each resulting gamete cell has a different mixture of alleles and mutations and is genetically unique, both from each other and from the original parental chromosomes in the original cell. This comes from both the crossing over that we mentioned before and the fact that the sister chromatids were randomly sorted into the daughter cells. This increases diversity in how DNA is passed from generation to generation. But fertilization increases genetic variation even more. In fertilization, two unique gametes from two different individuals come together. This keeps each new individual genetically unique. This incredible genetic shuffling, reassortment, and recombination results in genetic diversity and variation. So why is it important? Variation is important for healthy populations because having more genetic diversity and a large variety of alleles can help populations stay healthy by allowing them to adapt and respond to environmental pressures. So, for example, take this population of flowers that we have here. Some have an allele for orange color, some have an allele for yellow color, and some have an allele for purple color. If a whole bunch of beetles come in and they specifically eat only the purple flowers, having flowers around with other alleles that give rise to different colored traits allows some of those individuals to still be able to pass on their genes to future generations, helping the population adapt. And all that genetic variation comes from mutations and sexual reproduction.